Okay, so this is where we left off on. I believe we left off on talking about diastereomers, right? Okay, so remember that diastereoisomers are going to be stereoisomers where you essentially have two things that have changed. And so they're not going to be enantiomers, which are non-superimposable mirror images. They will in fact not be mirror images, but they will also not be superimposable, okay? And so you can see here, this is a very nice example where it's two sugars that we have D-altrose and then D-glucose. Yes, that's the glucose you need for your brain. Yeah, we can see two changes. See that? Where we flip two groups instead of just one. Okay. Notice that if we flipped all of the groups, that would actually give us the mirror image, right? Which would be the DL forms, right? Okay, now this right here is then going to be subdivided. There's epimers. This is going to differ at one of several asymmetric carbon atoms. Probably your most common example of this is actually your sugars. And so here again we see a D-glucose versus D-mannose. And we see that we've actually rotated again the same two groups, but we've rotated them differently this time, right? So that one of the groups has been changed right here. The other group has not changed from the glucose. You see that? Okay. And so only one of the groups has changed, and you've changed it at one of several asymmetric carbon atoms, these being the asymmetric carbon atoms right here. And so that is an epimer. Anomers, these are going to be isomers that differ at the new asymmetric carbon atom that's formed on ring closure. Again, I already did this, didn't I? This is like 99% of what the sugars actually look like. They're usually ring closed in your blood, okay? The vast majority of your population will be ring closed. The ring open version is just easy to memorize and easy to see what the differences are. So they get used a lot in books, but it's kind of a BS thing. Okay? Understand that. That's an entirely human perspective thing, not actually real world. So the common monosaccharides, again, these are going to be epimers, are going to be sugars that are diastereomers, that differing in the configuration only at a single asymmetric center. These then are the most common monosaccharides that you should have a pretty good idea of. Notice we actually start with D-ribose and D-deoxy-D-ribose. Those sound familiar? Yeah, if they had closed chains, they might be in some sort of RNA and DNA, wouldn't they? Good stuff. Okay. And so, again, what you should do here is you should have a pretty good understanding that you could identify one from the other. Okay? Now, the easy way to do that is not to memorize the whole thing. It's to memorize aldose versus ketose, and then understand how long it is, and then remember where you have the changeover, okay? The D versus L are actually defined on this last carbon. That's why it's green. Everybody see how it's green right there? Okay. What does the blue part here tell you? Exactly. Yeah, is this an aldose or ketose? If it has a hydrogen on the carbon, it's a aldehyde, right? If it does not have a hydrogen on that carbon, it's a ketone. Yeah, ketose, exactly. Okay. Again, here's the D-glucose. Anybody noticing a pattern for the Ds? This last OH group is on which side? The right side, exactly. That's the D, okay? D-mannose, D-galactose, D-fructose. Notice how they're arranged in here. It's arranged based on the number of carbons. Yeah. And that's a great way to go ahead and practice them and keep them in your head, is having them arranged by your number of carbons. Because it makes life easier when you're trying to identify them. You can basically go through a branch in your head, right? Okay. How many carbons are there? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. Okay, then this set is 6, this set is 5, right? Okay, so most monosaccharides will actually exist as interchanging cyclic forms. The aldehyde can react with an alcohol to form a hemiacetyl. Remember the hemiacetyl we've talked about in organic chemistry? I think I even pointed out that, hey, this is going to be really important in biochemistry because we're going to see a bunch of these. Good deal, huh? Finally paid off. All right, so what do you notice about these? These have many what types of groups? Alcohols, many alcohols, right? Okay. And so these very easily undergo these hemiacetyl closures. Again, this is the hemiacetyl right here. It's going to be the sugar's aldehyde in this case, reacting with the alcohol portion on the other side, making a hemiacetyl. The ketone can react with an alkyl, but it's going to form a hemiketyl. Okay, that's the only real difference there. They're the same sort of reaction. They're just one's a ketone, one's an aldehyde, right? Okay. And that means one has an OH on it, right there. They both have the OR. The big difference is H versus R right here, right? This makes it a hemiketyl. This makes it a hemiacetyl. Okay, so this is the pyranose formation. It's called pyranose because it has a similarity to pyran. That's pyran right there, by the way. So this is the D-glucose. That's the open chain form. We're then getting a ring closure reaction, which is this right here. So we can see here that this alcohol, remember alcohols are what? Nucleophiles. They're weak nucleophiles, but they are nucleophiles, aren't they? Okay. Notice how many carbons this is down. 1, 2, 3, 4, 5. That making a 6-membered ring, doesn't it? What's one of the most stable rings you can get? 6-membered rings. And so this has a great deal of stability, doesn't it? Yeah. And again, we have a carbonyl here, which is a very good electrophile, right? Carbon there. Okay, so it's going to attack the carbonyl. The electrons are going to form a bond. It will do that normal deprotonation. Remember where we had the proton still attached? And deprotonates. Okay? And that will give us these right here. Notice that we can get 2 different ones depending on which side the OH ends up on. Okay? And so if it comes from the top, we get that one, don't we? Comes from the bottom, we get that one. Now, we denote this with alpha or beta. Alpha is on the bottom of the line. It doesn't get boosted up to the top of the line, right? Because it's very small. And so alpha gives us the OH group on the bottom of the ring. Beta boosts the OH group up onto the top of the ring. Anybody have any question there? No? Note that this oxygen is in the ring. The alcohol oxygen is in the ring. The carbonyl is going to be the one above or below. Okay? Okay. All right. Anybody have any questions at all? No? Okay. Can you guys understand the alpha, beta, and remember that? Okay. So if the OH is down below the ring, it's alpha. Exactly. If it's above the ring, it's beta. Fantastic. This is really important because this is where you typically get branching. Okay? You may have noticed that we actually talked about that in the DNA way back when, right? Okay, so these are the furanose, formations. They look like furon. Notice, by the way, again, 6-membered ring, 5-membered ring. Okay. And so this is going to be D-fructose. It's, again, one that has the ketone here on the second carbon. And so when it does a ring closure here, you're going to end up with a 5-membered ring. Okay? Again, what's the other one that's really stable for rings? We had the 6-membered rings and the 5-membered rings, right? Okay. So those are the 2 rings you typically have to deal with. Okay? Almost all the rings you encounter in biological systems are going to be 5 or 6-membered rings because those are very stable. So here we call the, again, we will have the ketone here getting attacked by our alcohol. Again, depending on whether it comes in from the top or the bottom, that could give you different types. But because this one has a substituent, what ends up happening? You get less of the other. Okay? It's less likely to occur. And so what we get is the alpha D-fructofuranose right here. This is your cyclic form of the fructose. All of these are in equilibrium, though these equilibrium arrows are typically not in physiological systems. They would not be even. Okay? This population would be 90-something% in almost all these cases. The ring is by far and away the more stable one on almost all the cases. Let's talk a bit about the anomers of glucose. Again, the anomer is going to be a diastereoisomeric form of your sugar. This is going to form when a cyclic hemiacetyl is formed and an additional asymmetric center is created. And this is why we need these alphas and betas, by the way. What do you notice about this carbon? How many things does it have on it now that are different? We have 1, 2, 3, 4 different things. That means it's a asymmetric carbon, isn't it? Okay. Previously, in this case right here, how many different things did that carbon have on it? 3. That was not asymmetric, was it? So we actually create an additional center of asymmetry, meaning we have to somehow denote an additional isomer being formed, right? Okay, so in glucose, your C1, that's the anomeric carbon atom, is going to become a asymmetric center. This is going to form 2 ring structures that are possible. We have the alpha and the beta. So the alpha D-glucopyranose is going to be the hydroxyl group that's attached to the C1 on the opposite side of the ring as your C6. And we can actually see that up here. Okay, C1 here. See the C1? Okay. 2, 3, 4, 5, 6. This right here is the C6. Everybody see it okay? Fantastic. Okay. Okay. So again, remember the alpha, the C1 on the opposite side, the hydroxyl on the opposite side of the C6. Okay. The beta is going to have the C1 on the same side as the C6. And that's what you're seeing right here. See, that's the C6 right there. That's the hydroxyl group, and they're on the same side. Okay? Okay, does anybody have any questions? By usual format, you would write this C6 on top all the time. And that's why normally people would just go top bottom, because they would normally write this up. Okay? Because when you're looking for things, where do you typically put them? The top, right? Yeah. Okay, so the D-fructose is going to rapidly interchange between 4 distinct ring structures. You have your C2 is going to be the anomeric carbon atom, because again, that is the keto. The pyranose form is going to predominate in solution due to the reduced steric hindrances. The furanose form is going to predominate in fructose derivatives. And so here we see the different examples here. And again, here's the alpha D-fructofuranose. That would be the alpha one that we actually looked at right there. Right here would be the beta version of that. And again, the OH is on the top next to that one. See that okay? And then over here, we have the pyranose version of this. This is going to be ring closing with that alternative alcohol. Okay. Notice that this ends up on the same carbon as this. Okay. And this one typically is going to be shown in a fructose derivative. Okay. And again, this is the C6, but it's less energetically favorable because it has more stuff all stacked in the same area. Okay? Steric interferences, right? Okay, this is the beta, by the way. Everybody see it okay? Okay, so the most common monosaccharides exist primarily in the ring forms. Usually it's a pretty huge difference between the populations. These would be the ring forms of these various different ones, the most common ring forms. These are all alphas, by the way, down here. And these are the betas up here. Does anybody have any questions? Note, by the way, those are those ribonucleic acids that you've been seeing. That's that same ring form we've always been seeing, right? Okay, so the pyranose and the furanose rings, these are going to be assumed different conformations. The pyranose rings are not planar because of a tetrahedral geometry of its saturated carbon atoms. They can adopt two types of conformation. We have the boat and chair. You may remember the 6-member rings where we talked about boats and chairs. Okay. These can adopt, in the chair form, your substituents on the carbon ring atoms can be axial, nearly perpendicular, or equatorial, nearly parallel. The axial substituents will be sterically hinder each other if on the same side of the ring. Notice that's that same steric interference that we actually did calculations for in organic chemistry. If you kind of remember those, they had little tables, remember, where we looked at the different axial groups that could be on there. Okay. These are the chair and boat forms of your beta-D glucose. We have the chair form that's going to predominate because all axial positions are occupied by hydrogens. You can see that here. The boat form is disfavored because you have a steric hindrance where you have an overlap of the OH with, where you have axial OH overlapping with other things in the ring, right? See that? It has a nice overlap here. This causes hindrance. Again, you can see the boat, the chair. Y'all remember that okay, right? Anybody have any questions about this? Confusions? Okay, so the envelope conformations. Envelope is a structure that you see in the 5-membered rings. So the furanose rings, these are not going to be planar again. They're going to commonly adopt a conformation. This is called your envelope form. In the ribose moiety of most biomolecules, there's actually 2 common conformations, and those are those different endo positions. So we have the C2 endo, which is this one right here. And so the C2 position here is going to be out of your plane, this being the plane right here. Everybody see that okay? The C3 endo, excuse me. Oops, I pointed the wrong one, I think. I didn't really pay attention to my stupid cursor. Okay, so this right here, see how the color part is down here on the oxygen? And these, 1, 2, 3, 4, that's your plane. Okay, C2 right there. And that's the C2 endo. This one is the C3 endo. And again, that's your plane. That's the one that's endo out. That's your envelope. Okay? Everybody see it okay? Fantastic. Okay. All right. There we go. That should do that. So in D-glucose, this is going to be one of your most important fuels for most organisms. It's sort of the primary fuel for most organisms other than ATP. And so blood sugar, this is going to be your D-glucose in circulation in your blood. It's the only fuel used by your brain in non-starvation conditions. It's the only fuel used by your red blood cells in similar conditions. So the potential reasons why the D-glucose is an important fuel is that glucose is formed from the formaldehyde under prebiotic conditions. This may be available as a fuel source for primitive biochemical systems. The glucose is relatively inert. The most stable ring structure is beta-D glucopyranose, and so it's really stable fuel. It doesn't come undone and get oxidized on its own. And so those are pretty good reasons. They make a lot of sense if you start thinking about them, right? A little interesting side note to this is that a lot of the brain diseases are actually metabolic diseases. Okay. They're literally, your mitochondria or glucose is not working properly. This is pretty recent research, so if you're interested in that sort of stuff, you should look that up. It's really fascinating. The solutions of glucose, there's two endomeric forms. There's the alpha and beta. These are going to be in equilibrium. This will pass through the open chain form. And so you'll have a constant equilibrium between the alpha beta, where in solution, they open up spontaneously, and then they fold to one of the two, right? Spontaneously, okay? And you'll keep getting that exchange. If you happen to have one form that's more than the other, you'll have an exchange between the two. There's roughly a 2-to-1 ratio of your beta to alpha anomer conformations for D-glucose in an equilibrium solution because the beta is a bit more stable than the alpha, okay? And that gives you your overall populations of 2-to-1. In its linear form, glucose can react with oxidizing agents. Again, this is your linear form here. So one of the great examples of this is the linear glucose can react with your copper 2+, yielding copper 1+, and glucagonic acid. Again, this happens non-enzymatically with hemoglobin as well. So here you have the closed chain form. It can't react because it doesn't have the carbonyl available, right? There's no carbonyl, okay? However, when you have the open chain form, which it will have a small population that exists in, it can react with the copper 2+. And so the copper here, 2+, would go to copper 1+. This would actually go to copper 2 oxygen. And this reaction is going to involve water and hydroxide. And that's going to be an attack on here, making the carboxylic acid here. Well, the deprotonate carboxylic acid, the carboxylate. Notice that's that same reaction we looked at. Remember our copper-induced reactions? We made the carboxylates, usually to make something else, right? That's the exact same reaction. And that happens non-enzymatically. It can happen in your red blood cells. It can happen anywhere where it has copper concentrations that are pretty reasonable. And this is one of the reasons why this glucose right here is the predominant form in your blood. Because it has the highest percentage of time that it stays in the closed ring form. Okay? And that's not what we just mentioned, right? If you had something that had carbonyls that got exposed more often, what would happen to it? It would react more often, wouldn't it? Okay? Okay, so let's talk about reducing sugars. There's Failing solution. This is going to contain this solution of copper 2+. This is actually a test that's commonly used for the presence of sugars that adopt an open structure. Reducing sugars is the sugars that are going to react with oxidizing agents. All monosaccharides that can adopt linear structures in solution are going to be reducing sugars. Non-reducing sugars are going to be sugars that do not react with oxidizing agents. If it does not unfold, it would not be able to react, but also if it didn't have a carbonyl for some reason, it also would not react. Or if we block the carbonyl somehow. Glycation of sugars. So glycation is going to be the non-enzymatic addition of a carbohydrate to another molecule. Literally kind of like glucoseation, right? Glycation. Okay. This can be benign or detrimental. It can be on purpose or not on purpose, which is quite interesting, right? Okay. Normally, if you get sweeter, it's good. This is not a case where sweeter is better. The example is reducing sugars will non-specifically react with free amino groups on proteins. Often those amino groups would be like a lyse or arg. These will form a stable covalent bond, in which case you would have glycated or glycation of your enzyme or protein. The D-glucose has a low tendency to glycate proteins unless concentrations of sugars and proteins are very high for very long periods of time. Again, that's because it doesn't unfold very often, right? But, I mean, hell, if you have enough of it, then even 1% is a lot, right? Yeah. So even if you're only 0.4%-ish, which is usually most cases, if you have enough of both, it's going to happen, okay? But this is one of the reasons why if you're going to store your energy somehow, glucose is one of the best ones, right? What if you stored it in a sugar that instead of being like 0.4% was typically like a 5 or 10% unfolded? You'd have those reactions occurring much more often, right? Yeah. You'd start glycating more proteins. Okay, so advanced glycation in products, or AGESs, ages. Products are going to result from cross-linking following the primary modification. These have been implicated in aging, which the abbreviation is just amazing. The fact that they got that to work out. Wow. Really good job. Implicated in aging, arteriosclerosis, diabetes, other pathological conditions. Again, the name is so perfect, right? What does it do? Ages you. What is it called? Ages. Okay. So let's assess some of the treatments for diabetes mellitus by monitoring A1C levels. D-glucose will react with the hemoglobin to form glycosylated hemoglobin. This is the hemoglobin A1C. Sometimes the C is lowercase, sometimes it's capital. It just depends on really the program usually. Usually it's automated. And so whoever's doing it doesn't even choose. Whoever programmed it usually chooses. So this has no effect on O2 binding. In non-diabetic individuals, about less than 6% of the hemoglobin will be glycated. In patients with uncontrolled diabetes, almost 10% of the hemoglobin is glycated. So that's that difference in A1C levels that you typically see. The monosaccharides are going to be joined alcohols and amines through glycosidic linkages. The monosaccharides' biochemical properties can be modified by reactions with the following, which are alcohols, amines, phosphates. You're probably not super surprised about that if you remember any of your organic chemistry, right? This is literally just the list of common nucleophiles that reacted with carbonyls, right? So modifications are going to increase biochemical versatility. These can serve as signaling molecules. These can facilitate metabolism. So some of your glycosidic linkages, the O-glycosidic linkage is going to be a covalent linkage between the anomeric carbon atom of a carbohydrate and the oxygen atom of an alcohol. We actually see that down here. The N-glycosidic linkage is going to be the covalent linkage formed between the anomeric carbon atom of the carbohydrate and the nitrogen atom of an amine. Notice the N versus O. O meaning to oxygen, right? N meaning to nitrogen. Same terminology as organic chemistry, right? So it's still consistent. Okay, so again, this is your O-glycosidic bond. Notice this is an alpha linkage because it is below on the opposite side of this one, right? This one right here is an N-glycosidic bond. Again, notice 2 nitrogen, and it is a beta linkage because it is on the same side. Okay. Or another way of saying that, a buffering. Okay. Anybody have any questions about those? Anybody tell me what this is? Yeah. It's AMP, as in the AMP that might get put in your which one is this? RNA. It's not the deoxy form, right? Okay. Anyways. So the monosaccharides, this can be modified by the addition of substituents other than the hydroxyl groups. And so here we see some different examples of this. So here we have a CH3 that's been added to this. This one right here has a acetyl amine. Notice that would be the acetyl group right here attached to an amine. Okay. Right here we have the acetylglucosamine here. So again, this is another acetylamine attached to it. The big difference here being this is glucose, this is galactose. This right here is sialic acid, abbreviated SIA. This R group is this group right here. There's just not room for it. Okay. And then this is the acetyl group right here. That would be the acetyl right there. And then it's an amination because you've attached an amine right here. Okay. The phosphorylated sugars are going to be key intermediates in metabolism. So remember, phosphorylation is going to be the addition of phosphoryl groups. Very common modification of sugars in metabolic reactions. Your purposes of phosphorylation is going to be to make sugars anionic. This prevents crossing the lipid bilayer, and so you trap them there. They don't wander off. The interaction with the transports or the unmodified sugar. It'll block all of those. Blocks the formation of alternative ring conformations, and so you don't get different conformers that then won't fit in your enzyme, right? It creates reaction intermediates that more readily undergo metabolism. Remember that linking a phosphate typically makes it a higher energy form, right? And so now you have energy to do the next step of your reaction, okay? So really useful, right? Everybody understands why you need to trap it in place too? And how that works? Remember your lipid bilayers, they're neutral and they're hydrophobic, so they don't allow anything charged to go through, okay? Okay, so these are some different examples. These are some of the most important intermediates that you're going to see for the breakdown of glucose and your phosphorylated sugars here. So we have glucose 6-phosphate. This is abbreviated G6P. You'll see that a lot when we start getting more into the metabolism. Right here is dihydroxyacetone phosphate. This is DHAP. Everybody see that looks like the acetone right there is acetone. These would be the two hydroxys. That'll right there be hydroxy normally, but has a phosphate attached. Okay, this right here is glyceraldehyde 3-phosphate. That's GAP. Have you guys heard of these before? Nah. Yeah. You may have heard of them in like shuttles and stuff, or maybe in metabolism talk. We'll go into it in more detail. But introductions, say hi. You'll be your best friends in a while. Okay, can everybody see why this looks like glyceraldehyde, by the way? Glyceraldehyde has 3 OHs on it normally. Or glycerol, excuse me, has 3 OHs. Glyceraldehyde has an aldehyde here instead of an OH. And then we've attached a phosphate to it on the third position. Okay, so the monosaccharides are going to be linked to form complex carbohydrates. The oligosaccharides are going to be sugars that contain 2 or more monosaccharides. These are going to have to be linked by O glycosidic bonds. O meaning that you've used an oxygen to do the linkage, right? Which is good because the sugars don't have amines, do they? So they're going to have directionality. This will be defined by the reducing and non-reducing ends. The reducing end, again, is going to have a free anomeric carbon atom. These can be formed the open chain form. The non-reducing end is going to have an anomeric carbon and a glycosidic linkage. This cannot be converted to the open chain form, and so it can no longer do the reducing reactions we talked about before, right? This is the example right here, and it has everything nicely labeled. So here we have an alpha 1,4 glycosidic linkage. Again, notice glycosidic linkage is always an O linkage, right? Oxygen linkage. We're the alpha. Can everybody tell me why we're alpha? Yeah, it's the one is the one below or on the opposite side, right? Yeah. And then the 4 right here is this group right here. Okay? Okay? And this would normally be your reducing end, but it can't open anymore, can it? Because we're stuck here. We've been bonded. We can't do anything with it. And so what's going to end up happening is this will end up being your only reducing end. It's the only one that could do a ring opening, right? And when it does the ring opening, it would expose the aldehyde or ketone, depending on the sugar. And that would then be the one that could do the reducing, right? Okay, does anybody have any questions here? Okay. Everybody's comfortable with the numbering? Okay. 1 here. 1, 2, 3, 4. So sucrose, lactose, and maltose, these are going to be common disaccharides. Disaccharides, di meaning 2, right? And so these are 2 monomers of the saccharides, or disaccharide 2. These are going to have 2 sugars. These are going to be joined by an O-glycosidic linkage, and here we see some examples. The cleavage products of the disaccharides can be processed to provide energy in the form of ATP. And again, here we have the common name for these, which is sucrose. This is sucrose right here, in case you're ever wondering. Okay, and this is going to be an alpha D glucopyranosyl in a 1,2 linkage. Can everybody see the 1,2 linkage here? Okay, fantastic. This is then to beta D fructofuranose. Okay, yes, that would be fructose right there. And that is sucrose. This is alpha lactose. Anybody know of lactose? It's in like lactose, sugar in milk that makes you sick if you have lactose. Okay. Anyways, this right here is it right here. We can see it's galactose with glucose. Okay. Notice again that's a beta linkage here. You note the weird wavy thing? That's because this one's down here and this one's up here. And so if they want to put it on one line, they have to do that. But normally it's represented with this being boosted onto another line. Okay? Anyways, this is maltose. Anybody know maltose? You ever had a malt ball? That's that flavoring. Yeah, maltose gives you the malt flavor. Anyways, it's 2 glucoses. So the disaccharide sucrose. Sucrose can be a disaccharide of sugar cane or sugar beets. This will consist of glucose linked to fructose. The anomeric carbon of glucose is going to be linked to the anomeric carbon of fructose, which actually creates a kind of interesting thing that you haven't seen before this, because you have the alpha and beta at the same site, right? The configuration for the alpha for glucose, it'll be beta for fructose. It's not a reducing sugar at all because we've used the reducing end on both sides, haven't we? Okay? And so it can't do a ring opening on either side. This can be cleaved by sucrase, which is another name for that is invertase. So lactose, another disaccharide. This is the disaccharide that you usually consider with milk, of course, lactose intolerance. This is going to consist of galactose linked to glucose. It's linked by a beta 1,4 glycosidic linkage. Can be hydrolyzed by lactase in the human beings and beta galactosidase, in bacteria. Maltose or maltose is going to be a disaccharide that's resulting from the hydrolysis of a large oligosaccharides that consist of 2 linked glucose molecules. It's joined by an alpha 1,4 glycosidic linkage, can be hydrolyzed by 2-glucose by maltase, which is another name for maltase is the alpha glucosidase. Typically happens due to digesting much larger polysaccharides. Okay? So maltase inhibitors can help to maintain blood glucose homeostasis. Homeostasis? Man, my mouth is just... Okay. Maltase inhibitors can help to maintain blood glucose homeostasis. Man, I need to start doing singing practice before this. I need to teach this first before the other class from now on. So after a meal, we have starch and glycogen. These are going to be degraded by alpha amylase. Oligosaccharides will be generated by the alpha amylase and are going to be further digested by the maltase, again, ACE being the enzyme, right, that will act on maltose. The acarbose, or precose, that's this one right here, by the way, and migalitol, and glycet was the other name for that, which is right there, are actually competitive inhibitors for the maltase. And so human milk oligosaccharides is going to protect newborns from infection. You've all probably heard of that, where if you have a newborn actually be fed breast milk, especially directly from the mother, they'll have a much better immune system. Okay. So there's more than 150 different oligosaccharides that have been identified in human milk, not digested by breastfed infants. They play a significant role in protecting them against bacterial infection. One of the infections is Streptococcus bacteria that may be transmitted during vaginal childbirth. There's a really interesting series of studies having to do with childbirths, and apparently traveling through the placental actually activates certain things in the child to develop properly. And so if they don't have that placental put on them when they're born, they actually don't develop properly. Their immune system doesn't. And the breast milk also then activates their immune system, which is, it's just insane. Anyways, who the hell would have known? So the oligosaccharides may serve as a fuel source for beneficial bacteria. They prevent the attachment of microbial pathogens to the intestinal wall of the newborn. If you do not have the placental and the human breast milk for the baby, by the way, it will not develop a proper biome in their gut. And they'll be much more prone to autoimmune infections, everything. It's insane. Yeah. So, anyways. By the way, in case you're ever wondering why human breast milk is like a premium item in the black market, yeah, people do that for after they're born too. And apparently it's supposed to help. I don't know if there's any data on that. It's weird. It's weird. Anyways, glycogen and starch is going to be storage forms of your glucose. The free glucose, this cannot be stored because you have high concentrations are going to disturb your cell's osmotic balance. It would also start causing reductions, right? You start reacting with proteins. So you can't store it as free glucose, right? Your cells would burst or they'd get their proteins reacted with. So your polysaccharide glycans are going to be large polymeric oligosaccharides. It's going to be formed by the linkage of multiple monosaccharides. These will play roles in energy storage, structural integrity. Homopolymer is a polymer in which all the monosaccharide units are the same. Homo meaning same, polymer, right? So repeats of the same saccharide. Anybody know glycogen or starch? You've probably heard of those, hopefully. Okay. What do you typically find the glycogen for? Like animals, right? Starches. Yeah, usually plants, right? That's more the plant one. Okay, so glycogen, this can be large branch polymer of glucose residues, most common in homopolymer in animal cells. It's the homopolymer we typically use. It's the majority of our homopolymer. Okay? This is going to be storage that's form of the glucose, excuse me. Man. Okay. Storage form of glucose. Most glucose units are going to be linked by an alpha-1,4 glycosidic linkage. Right there you can see that. That is your quote-unquote straight chains. Okay? You'll form branches by the alpha-1,6 glycosidic linkages, which you see an example of right there. It's going to hydrolyze by alpha amylase. The branching is going to increase your surface area. This will allow better access for enzymes to rapidly break down the glycogen. It also tends to make it a bit more dense, so it can more easily store. But no, the biggest thing is we can access it fast. Do you know where the majority of where this is stored in the human body? Anybody ever heard of a beer gut? That's a beer gut. Okay? Because the beers have a lot of glucose, sugars, and so they get converted if it's not glucose into glucose. And then they get assembled into glycogen, right? And then they typically build up around your liver where you have the enzymes to quickly hydrolyze them back into glucose. We'll talk about that more later though. Starch is going to be the homopolymer that serves as the nutritional reservoir, typically in plants. There's actually two forms of starch. There's amylose and amylopectin. Okay? The amylose is going to be the on-branch type of starch. This can be composed of glucose residues and the alpha-1,4 linkage. You may notice a similarity. Alpha-1,4 linkage, same one. Okay? Okay, the amylopectin, this is going to be a branch type of starch with approximately one alpha-1,6 linkage, i.e. one branch, for about 30 alpha-1,4 linkages. Okay? And it's going to have an identical structure to glycogen. The only difference is it's a lot more straight chain, a lot less branching. Okay? The amylose and amylopectin is going to typically be hydrolyzed by alpha amylase. Again, same stuff. Does that make sense to everybody? Are plants in a hurry? No. Not typically, right? So cellulose is actually the main structural polysaccharide of plants. Cellulose is going to be the on-branch polymer of, again, glucose residues. These are going to be joined by beta-1,4 linkages. It's going to serve a structural role instead of a nutritional role, though. Well, in plants. In certain types of bacteria and fungi, it's going to serve a nutritional role. Or deer, right? The beta configuration is going to allow the cellulose to form long, straight chains. These are going to interact with one another through hydrogen bonds. This yields a very rigid, or at least for its weight, a very rigid supportive structure. The alpha linkages of starch and glycogen are going to form compact, hollow cylinders. These will be suitable for accessible storage. Okay. And so essentially it's just the beta-1,4 linkages and cellulose, which we don't have a picture of here. I guess I forgot to put a picture of here, but you can imagine it. That is the alpha-1,4. Beta-1,4 would be the other way. Okay. Okay. And it makes very, you've seen wood split, right? It'll come apart, paper come apart. It makes fibers that stick together pretty well. And that's what gives that cellulose its structural component. Oh, there it is. Of course. Okay. And so, God, sometimes I forget where I put my fucking fingers. Okay, so here we have our glycosidic linkage. These are our beta-1,4s, by the way. See our beta-1,4? And you can see this much better because we also have the chair configurations. See how that chair configuration alongside the beta-1,4 gives you really nice straight fiber chains, right? That you can then weave together, which is literally what you're doing when you're making rope, isn't it? You're weaving together cellulose fibers, okay? Same thing when you're making cotton, same thing when you're making bark rope, any of that stuff, hemp rope, anything, okay? This is actually made out of cellulose, by the way. This plastic-looking cup here. Yeah, these are just cellulose fibers laid out into a weave, essentially. Yeah. Anyways, this is the starch and glycogen, and you can see here it actually kind of curves. It doesn't do a straight. Can you see that okay? And it makes more sense once you start looking at this chair configuration, right? Because it can't be straight because of the chair configuration. Okay? Okay, so insoluble and soluble fiber are going to be important parts of our diets. Mammals cannot digest cellulose because we lack cellulases, but plant fibers are still going to be important to the mammalian diet. Insoluble fibers are going to increase the rate at which digestion products pass through the large intestine. Typically, it's just like, it just squeezes everything else through. Okay? This will soften stools and make them easier to pass. The soluble fibers, on the other hand, like pectin, or polygalacturonic acid, is going to slow the movement of food through the gastrointestinal tract. This is going to facilitate absorption of nutrients from the diet. What time is it? Time? Okay. I'll go and quit here. We'll go into chitin next. Did anybody get insoluble versus insoluble fibers? Okay. You all have a wonderful day. And man, we are really close, aren't we? Yep. Next time we'll finish it, and then we'll be on to the testing.